So hello, my name is Nicole King. I'm an investigator in the Howard Hughes Medical Institute and a professor at the University of California, Berkeley. And I'm excited to be here today to tell you about organisms that we study in my laboratory, the choanoflagellates, and tell you about how they interact with bacteria and how these interactions might inform us about animal origins. Now, I want to provide a little bit of introduction to the motivation for the research in my lab. There's been a lot of focus in the past on understanding how different animal body forms diversified and understanding how different animals are related to each other on the phylogenetic tree. But in fact, we know relatively little about the nature of the organisms from which animals first evolved. And in my laboratory, we're particularly interested in understanding the genomic innovations and the influences of cell biology and interspecies interactions, and understanding how that might have contributed to what we call the transition to multicellularity. That is, how did ancestrally unicellular organisms evolve into organisms that are capable of simple multicellularity, such as this hypothetical colony? In part one of my talk, I previously spoke about how an unusual group of organisms called the choanoflagellates can help us understand animal origins. And I told you that the first animals likely had genes in their genomes that had evolved much earlier. And so choanoflagellates are, by comparing choanoflagellates to animals, we're learning about the nature of the first animals. In this part of my talk, I'm going to focus on one particular species of choanoflagellate. And I'm going to tell you that this choanoflagellate actually can transition from being single-celled to having simple multicellularity. And I'm going to tell you about how we've been developing this into a new model system so that we can learn about how that transition from being single-celled to multicellular is regulated. And what we don't know now, but we hope to learn, is whether the regulation of multicellularity in this organism might give us specific insights into the ancestry of multicellularity in animals. Now, the organisms that we study are in fact not animals. They are the sister group of animals. They are called choanoflagellates, and they sit on this very special part of the phylogenetic tree because they are the closest living relatives of animals. And our understanding that choanoflagellates sit here on the tree and that they are our essentially evolutionary cousins comes from multiple lines of evidence. It comes from comparisons of the cell biology of choanoflagellates to the cell biology of animals. It comes from many different independent types of phylogenetic analyses, and it comes from comparisons between the genomes of choanoflagellates and the genomes of animals. From all of these different sets of data, it's now become very clear that the study of choanoflagellates, our closest living relatives, tells us about the biology of our last common ancestor. So I introduced this in part one, but I just want to quickly review the biology of choanoflagellates because it becomes essential for understanding what I'm about to tell you in the later part of this talk. Choanoflagellates are microbial eukaryotes. They have a spheroid or spherical or ovoid cell body, an apical collar of actin-filled microvilli, and a long apical flagellum. And this flagellum can undulate from side to side. And this undulation creates water currents that allow choanoflagellates to swim through the water column. But these water currents also pull water from the media up against the collar. And those water currents can carry bacteria. And bacteria are actually the primary prey target for choanoflagellates. Choanoflagellates are voracious bacteriivores. They love eating bacteria. They're very good at it. And this is an essential part of their biology, their ability to capture and ingest bacteria. So choanoflagellates have a lot of really interesting aspects to their biology, one of which obviously is this ability to eat bacteria. But one of the aspects of their biology which really excited me when I first learned about it as a postdoc is that some choanoflagellates can form these beautiful multi-celled colonies. These colonies are, in fact, to me, reminiscent of some types of marine invertebrate embryos. And these colonies raise all sorts of questions about how do the cells interact and how is this process regulated. Moreover, you'll remember that I told you that one of the major questions in my laboratory is how evolutionarily did the ancestors of animals evolve the ability to form simple multicellular morphologies. And here we have, in living color, an organism that actually does it every day. And so what we've been doing in my laboratory is to understand, or is to study this process in minute detail, the same way that a Drosophila biologist might try to study how a fruit fly goes from an egg to an embryo to an adult, or a mouse biologist studies the same process of development. We're trying to study in mechanistic detail the process by which this organism, S. rosetta, goes from being a single cell to a multi-celled colony. And in particular, we're focusing on trying to understand the mechanisms of cell adhesion and cell signaling within the colony. We're trying to understand what triggers this transition from being single-celled to colonial. And eventually, we hope to learn whether the mechanisms underlying this transition in choanoflagellates are related to mechanisms underlying animal development, in which case we would infer that they are ancient and evolved before the origin and diversification of animals. So I'm going to tell you a lot about this organism, S. rosetta. And the first thing I need to tell you is that it does a lot of exciting things. You know, I think many of us think about protozoa as being simple organisms that lead a rather mundane life, but this organism not only can switch between being single-celled and colonial, it actually has a really wild and crazy life history. It has many different morphologies that it can produce, and these are all coming from an organism. You know, a single genotype is encoding for all of these different forms, and many of these forms can differentiate into other forms. So this cell in the center we call the slow swimmer, and cultures that only have slow swimmers in them are capable of producing rosette colonies, chain colonies, fast swimmer cells, and these fast swimmers can differentiate into these attached cells. So there's a lot of dynamic cell differentiation that's going on. And it seems to have at least some environmental component because we can, in the laboratory, push the choanoflagellate toward different types of cells by changing the environmental conditions in which we grow the cells. For the sake of simplicity and also to focus on things that we know the most about, today I'm only going to focus on this part of the life history and try to understand what is it that allows this cell to differentiate into these other multi-celled forms. In particular, we're going to talk about the rosette form because this is the form in which S. rosetta was actually isolated from nature, and this is the one that is most similar to the multicellular form that we hypothesize was required for the ancestry of animals. So we want to know how do rosettes form? Are they forming from multiple cells swimming together and sticking? And that would be similar to the slime mold, Dictyostelium. Or are they more similar to animals in the way in which they form? That is to say, does a single cell divide repeatedly to form a rosette? We'd also like to know how are the cells inside of the rosette adhering to each other? How do you get that stable structure? Again, trying to draw analogies to embryogenesis. And finally, we have this enigma, which is that this single cell is capable of producing 3 different types of morphologies. It can divide to produce more copies of itself. It can produce these chains, or it can produce rosettes. And we'd like to know how is that differentiation process regulated. So let me start with question number 1. How are these rosettes forming? To investigate this, we used multiple different approaches, but I think the simplest one is to just watch. And what we found is that when cultures were shifting from having only single-celled individuals to rosettes, it always happens through cell division. And so what you're going to see in this movie here is that this is a founding cell that's going to divide repeatedly to produce a spherical multi-celled colony. So let's watch. The single cell divides over and over again. The cells remain attached. And in the end of this 15-hour movie, we have a spherical colony. And this, I think, is also nicely shown here in these stills taken by confocal microscopy. Now, I want to make the point that even though this looks two-dimensional, these colonies are actually three-dimensional and producing a nice sphere. Okay, so the answer to our first question then is that the rosettes are forming through cell division, and that provides a very nice parallel to the way in which embryos of animals form, in which you have a single cell, the zygote, and that zygote divides over and over again to produce a multicellular embryo. How are the cells in choanoflagellate colonies actually sticking together? And to answer this, we had to use electron microscopy. If you look at choanoflagellate cells using a scanning electron microscope, what you see is that the cells are actually connected by these fine intercellular bridges that you can see here. And we think, although we don't know yet, but we think that these are the product of incomplete cytokinesis. That is to say that the cleavage plane that forms when cells are dividing doesn't close completely. And so there's a little remnant of membrane that remains between those cells, and it produces this intercellular bridge. But that's not the only source of cell adhesion between these cells. If you examine the cells under different conditions and look at them either in SEM or in TEM, what you can see is that there is a fine meshwork of material covering the cells and also filling the inside of the colony. And this is actually extracellular matrix, or ECM. And so it's the combination of the intracellular bridges and intercellular bridge, and extracellular matrix that is contributing to the structural integrity of the rosette. Okay, so just to summarize then, I've just told you that when choanoflagellates form, when S. rosetta forms rosette colonies, it forms it through incomplete cytokinesis. And what I've also told you is that the cells in these rosettes are adhering through a combination of intercellular bridges and extracellular matrix. But of course, I think the question we should all be interested in and wondering about is, how is this transition regulated? And what determines whether this single-celled form of S. rosetta divides to produce more of itself, or produces chains, or produces rosettes? What is determining the regulation of this developmental switch? And here's where I was really stymied in my research. And so what I need to tell you is a story of frustration that finally ended with serendipity and I think an exciting new discovery. So let me back up and tell you about how I started studying S. rosetta. When I began my postdoc, there were no labs out there that were studying choanoflagellates. And so I was fortunate to be taken into the lab of a leading Evo-Devo researcher, Sean Carroll. But I had to go to the ATCEC, the American Type Culture Collection, to work with a protostologist named Tom Neerad to learn how to study choanoflagellates. And while I was there, he and a group of other scientists were studying diverse microbial eukaryotes from the environment. And he observed one choanoflagellate that was capable of forming beautiful rosette colonies. And this is S. rosetta. So he was kind enough to put S. rosetta into culture. He isolated a single colony, grew it up, and froze it down so that I could study S. rosetta in perpetuity. So I brought it back to Madison, where I was doing my postdoc, and started growing this choanoflagellate. And let me tell you, it was a very frustrating finding when I brought it back to Madison, because S. rosetta cultures in the laboratory rarely had rosettes. They were largely unicellular. And so you can see here would be a best case scenario, lots of single-celled choanoflagellates. You can see the round cells, and only the occasional rosette colony, and lots of bacteria. And so no matter what I did, these cultures would not robustly form rosette colonies. And so that meant that I wasn't going to be able to do the types of experiments that I wanted to do to study the mechanisms of rosette development. So I worked on this for a long time without success, and then ended up bringing, fortunately other things worked, but S. rosetta was recalcitrant. And so I brought it with me when I started my own lab at Berkeley and continued to experience frustration and continued to be unable to get this thing to form rosettes in any robust or predictable way. And so finally, I switched my research objectives and decided that if I couldn't get rosette colonies from this species, at least I could sequence its genome, and that might tell me something by comparing its genome to genomes of single-celled choanoflagellates, might tell me something about the mechanisms regulating development. And so an undergraduate in the lab at the time, Rick Zuzo, helped me to get this choanoflagellate ready for genome sequencing. And one challenge I need to point out is that because choanoflagellates eat bacteria, this creates a real problem for genome sequencing because the bacterial DNA can make it difficult to get a high-quality genome assembly from the choanoflagellate. So the first thing he had to do then was to treat these cultures with cocktails of antibiotics. And he tried 2 different cocktails of antibiotics, and they gave 2 very interesting results. So one cocktail of antibiotics, actually, when he used that, it resulted in a culture that had a bloom of rosette development. And so you can imagine, we were thrilled. It was so exciting, and we thought, we had no idea why this treatment with antibiotics led to rosette development, but it did. Perhaps even more interestingly, when he treated with a different cocktail of antibiotics, he recovered a culture that produced no rosettes ever. And so we found that different cocktails of antibiotics led to different results. And we started to wonder what was going on. Now, it could have been any of a number of possible explanations. It could have been that the antibiotics were directly stressing the choanoflagellates in different ways. It could be that the choanoflagellates were starving when exposed to one set of antibiotics, but not the other. But it was hard to reconcile these different observations. But the one possible explanation that sort of was consistent with what we were seeing was the possibility that bacteria from the environment were actually regulating the switch to rosette development. And so to test that, Rick took bacteria from the original environmental sample and added them to this culture that didn't form rosettes and asked whether those environmental bacteria could stimulate rosette development in these non-rosette forming cultures. And in fact, that did work. So environmental bacteria were sufficient to induce rosette development. So that was very exciting, very unexpected. And of course, the next thing we wanted to know was which bacteria were actually providing this stimulus for rosette development. And so what Rick and other members of the lab did was to go into this original environmental sample and isolate multiple independent strains of bacteria and test them one at a time in this rosette deficient culture and ask whether those bacteria were capable of inducing rosette development. And so we tested 64 different environmental isolates one at a time. And what we found in the end was that of all of these, only one species was capable of inducing rosette development. So what was that species? It was the previously undescribed bacterial species Algoraphagus machpanganensis. So we can add Algoraphagus to cultures of single-celled choanoflagellates, and that is sufficient to induce them to form rosette colonies. I need to make a couple of important points about Algoraphagus. First of all, it was co-isolated with S. rosetta, so it's a natural environmental cohabitant with S. rosetta, and it's also a sufficient prey target. So we can grow S. rosetta only in the presence of Algoraphagus, and it's perfectly viable, and it happily forms rosette colonies. The other exciting and interesting thing about Algoraphagus is that it's a member of a much larger group of bacteria called the Bacteroidetes. And Bacteroidetes bacteria are some of the most abundant bacteria in your gut. And they're also abundant and important bacteria in diverse environmental settings, including the oceans and soil. And in each of these settings, there's a growing interest in the ways in which bacteria might be influencing the biology of eukaryotes with which they're associated. So we're excited about the possibility that this interaction that I've just described to you might be used to help understand the mechanisms underlying interactions between bacteria and eukaryotes. Now, why would we be so excited about bacteria? I mean, I've hinted at that a little bit, but I want to back up and give you a little bit of context for why bacteria are such an important factor to try to investigate when you're thinking about animal origins. And to do that, I need to go in the way back machine. I need to remind you about the history of life on Earth. And to do that, I'm going to use this time chart in which we're thinking about life, the history of life, starting with the present here at the top, going back to the start of Earth and the solidification of the crust, and say that we think that the last universal common ancestor of life lived on the order of over 3 billion years ago. And the earliest fossil evidence we have for life is that of stromatolites. These are large multicellular aggregations of bacteria. They are essentially bacterial biofilm. And we preserve representatives of these types of morphologies here today. Here's an example of a modern stromatolite. And these complex forms are produced by bacteria. And there's a lot of really terrific work that's being done to address the ways in which bacterial metabolism has influenced the geochemistry of Earth, but also the life of other organisms. And what we now realize is that animals whose fossils have not been recovered, the oldest animal fossils are no older than about 500 to 600 million years old, and the oldest multicellular eukaryotes in general on the order of 1 billion years old. Those multicellular eukaryotes evolved in environments that were already dominated by teeming hordes of bacteria. If we're going to understand the origin of multicellular eukaryotes, we need to understand how their progenitors coped with a world that was already populated and colonized by bacteria. So that's one important point that I want to make about the bacterial context of animal origins. The second point I want to make harkens back to something I talked about in part 1, and that is that through the study of choanoflagellates, we've been able to reconstruct some important aspects of the biology of the first animals. And so you may remember that I mentioned that we think the first animals probably had collar cells, and more importantly, that those first animals were actually involved in bacterivory. That is to say, they ate bacteria. They made a living by eating bacteria. And so we now think that interactions with bacteria were an obligate part of the life history of the first animals. The final point that I want to make is that if you look at living animals, what you can see is that development in many of these diverse organisms is regulated by bacterial signals. The challenge in these cases, now obviously there's been a lot of interest, but the challenge has been that we're looking at large, complex, multicellular organisms that are growing in association with diverse and complex communities of microbiota. And this has made it very difficult to try to learn something about the mechanisms underlying these important interactions. And so what we are now doing is using this interaction between the bacterium Algoriphagus and the choanoflagellate S. rosetta as a simple bioassay to discover bacterial signaling molecules that we think will help us understand the regulation of this developmental switch, but will potentially have relevance to other systems as well. Now I have to say that this has been a very exciting but also challenging process, in part because we had absolutely no idea what the nature of the signaling molecules were. After casting about in the dark for a little while, we had a hint that came from looking at what is unusual about the Bacteroidetes, which are the bacteria, the large group of bacteria of which Algoriphagus is a member. So Bacteroidetes actually have, like other members of this group, an outer membrane and an inner membrane. They have components called LPS and peptidoglycan, which are known inducers of immune system components in animals. But they also have an unusual group of lipids called the sphingolipids and the closely related sulfonolipids. And I say that these are unusual, but in fact, they're quite common in eukaryotes. It's in bacteria in which you don't often see these types of lipids. And so for a variety of reasons, we focused on this group of lipids as a potential source of the signaling activity. And to do this, we have established really one of the best collaborations in my career. It's been a fantastic experience. We've been collaborating with John Clardy, who's at Harvard Medical School and has done fantastic work in many systems in recovering bioactive molecules. And so what he and his group did is they took Algoriphagus, they extracted the sphingolipid fraction from its outer membrane, and then these sphingolipids are very difficult to deal with. So at our first pass, we've now started using different approaches, but in the first pass, people from his lab used a process called PrEP-TLC, and this is thin layer chromatography, to separate out all those sphingolipids. And then they would scrape them off of this plate and send them to my lab, where we would test them in the bioassay and see whether those fractions were capable of inducing rosettes or not. And based on that, then we could take the fractions that were capable of inducing and analyze them by mass spectroscopy. And so this was an iterative process. We would send them bacterial samples. They would fractionate them. They would send us the fractions. We would test them. We would send them the information. It was back and forth. And through a long series of analyses, we eventually were able to identify the first bacterial molecule that was capable of inducing rosette development. And that is this molecule. We've named it RIF1 for rosette-inducing factor 1. And we now have a structure for it, which is very exciting. And we also know something about its chemistry. So RIF1 is not a sphingolipid. It is, in fact, a different class of molecules called the sulfonolipids. And the sulfonolipids differ from sphingolipids in that they have a sulfonic acid head group at one end. This class of molecules has not previously been shown to be involved in signaling. So this is exciting because it's the tip of the iceberg. These types of molecules might have wide-ranging roles, and we can just start to study them now. What is known is that in bacteria, they seem to have a role in regulating gliding motility. So this molecule we can fractionate and isolate from bacteria, but is actually functioning in a way that is consistent with it having a real role in the environment. And so we took purified RIF1 and tried to determine the concentration of the molecule that was required to induce rosette development. And the exciting result is that, in fact, RIF1 is tremendously potent. It's able to induce rosette development. Here, again, I'm showing you the extent of rosette development along the y-axis and the concentration of RIF1 along the x-axis. And what I hope you can see is that we are getting maximal induction of rosette development at concentrations that are in the femtomolar range. And so this, not only is it active at these levels, but these are the levels in which we find RIF1 in the conditioned media. And so the activity of RIF1 is entirely consistent with it having an important function in environmental concentrations. So that was very exciting. So it's a new class of signaling molecule. It's active at environmentally relevant concentrations. That's all good. But now we get to the nitty-gritty. I want to show you that, in fact, the maximal induction that we're seeing is only on the order of about 5% of cells going into rosettes. So it suggests that RIF1 is important. It's sufficient for rosette induction, but it's not the whole story. And so what we've now done is go back to our simple bioassay to see now if we can more rapidly discover other potential bacterial signaling molecules. And in fact, we have. So again, this is through our collaboration with the Clardy Lab. We've gone back now and analyzed more broadly, not just the sphingolipids, but the entire lipid fraction. And we've been able, through this process, to find other bioactive signaling molecules. Here in this part of the eluate, we find RIF1, but also many, many other sulfonolipids, all of which are inactive. And that makes an important point. RIF1 is inactive because it's a sulfonolipid. RIF1 is a special sulfonolipid. Most other sulfonolipids can't induce. So there's a tight structure-activity relationship between RIF1 and its ability to regulate rosette development. But what we've also found is that there are other classes of sulfonolipids that are structurally similar to RIF1 that are also capable of inducing. There's an entirely different class of lipids called the LPEs, or the lysophosphatidylethanolamides. These are able to synergize with RIF1 and the other sulfonolipids to promote rosette development. And finally, there's another type of lipid that's an antagonist of the RIFs. And if you incubate it with the choanoflagellate and then add RIF 1, 2, or 3, you find that you block rosette development. So there are many different diverse bioactive lipids available in Algoriphagus, and we can mix them together and reconstitute the full induction activity that you normally get from live Algoriphagus bacteria. So there are diverse bioactive molecules that we've discovered already using our bioassay just from Algoriphagus. But in addition, we've also surveyed lots of other diverse bacteria, and biologists will do this. And what I'm going to tell you is I'm showing you a phylogenetic tree of diverse bacteria, and you don't need to worry about the fact that you can't read which bacteria I'm showing you. The point I want to make is that there's a lot of diverse bacteria we've tested now, and shown in these squares, I'm indicating whether they induce rosettes or not. Those shown with black do induce, those with white don't, and those in gray induce at a low level. And so we're finding that across the tree of bacterial diversity, we're finding many different bacteria that induce, and some of them seem to use different types of bioactive molecules to induce rosette development. Finally, we'd like to know whether this bioassay might help us find something that's of biomedical relevance. And so we've actually surveyed the bacteria of the vertebrate gut system, looking at different parts of the intestinal tract and testing whether they're capable of inducing rosette development. And we find, in fact, that they can. So bacteria from the stomach and the small intestine do not induce rosette development, but bacteria from the cecum and the colon do. And if we follow the strategy that we followed previously in the discovery of Algoriphagus, we can do it here and culture these bacteria and see if we can identify the species that induce rosette development. And so we've done that, and we've now discovered the specific bacteria from the gut system that are capable of inducing rosette development, and we're focusing on isolating the bioactive molecules from these organisms as well. Okay, so let me just recap what I've told you. I've told you that rosette development is regulated as the process of incomplete cytokinesis, and cells in rosettes are held together through a combination of intracellular bridges and ECM. Moreover, what we've discovered, and it was quite unexpected, we've found that the developmental switch that controls whether a single cell is going to form a rosette, chains, colonies, or another single cell, that's regulated not solely by genetics of choanoflagellate, but actually, importantly, by signals that are released by environmental bacteria. So, over part 1 and part 2, I've been telling you about these really interesting organisms, the choanoflagellates, that were discovered in the 1800s, that we've now brought into the molecular and genomic era. And through the study of choanoflagellates, we're finding that we're able to reconstruct the biology of the first animals in increasing resolution. And one of the most exciting things I think we found through these types of studies is that many of the genes that are essential for regulating cell-cell interactions in animals and regulating the process of development actually evolved before the origin of animals and are conserved in the genomes of living choanoflagellates. So that's been great to learn that choanoflagellates provide this window into animal origins. In addition, I told you about a transition to multicellularity that actually happens in the life history of a living choanoflagellate, the Choanoflagellate S. rosetta. And the very exciting discovery that we've made by studying this process in mechanistic detail is that the developmental switch to form multicelled rosette colonies is actually regulated by environmental bacteria. So it's been very exciting. We've been collaborating with John Clardy to uncover bioactive molecules, and this now brings us to the point in which we can start thinking about the choanoflagellate side of the story. How is it that choanoflagellates are actually sensing these bacterial signaling molecules? Moreover, we're curious about whether this interaction is something special to the choanoflagellate lineage, or whether it actually is informative about mechanisms underlying animal origins. And so to that end, in the long run, we'd like to know whether the mechanisms regulating this signaling interaction between bacteria and choanoflagellates might be conserved in the interactions between animals and their commensal bacteria. So it's been a real pleasure telling you about this work, and I want to take this opportunity to thank a number of people, many of which, I can't list all of them, but I really want to thank everybody in my lab. And I've highlighted here in yellow the people who have actually contributed to the work that I discussed here. One current member, Ariel Woznicka, and many different alumni who have been essential to this project. Moreover, I have to express my gratitude to John Clardy, who's been a really fantastic collaborator, and I've learned so much from him, and it's been a wonderful experience. And then, of course, our funding agencies. If you find yourself fascinated by choanoflagellates and you want to learn more, we invite many, many people. We want to grow this community. And you can learn more about choanoflagellates in these various locations. And importantly, we have a choanoflagellate workshop every 2 years. So please come and join us. And it's been a pleasure talking to you.